Students, today we are going to study microtubules and microfilaments, which, along with actin and intermediate filaments, constitute a dynamic cytoskeleton. For your information, I should tell you that the cell also requires skeletal elements to maintain its structure and other functions like transport of vesicles and other material inside the cell. Microtubules were first discovered by D. Robertis. They are around 200 angstrom in diameter and are made up of 13 protofilaments. Each protofilament is having a diameter of 50 angstrom, and the space between inside the 13 arranged protofilaments is having a diameter of around 150 angstrom. So 150 along with 50 angstrom protofilament constitutes 200 angstrom size. They are made up of tubulin proteins. Tubulin protein is a dimer made up of alpha and beta tubulins, and whenever they polymerizes, that means alpha and beta tubulin protein is formed, and upon their depolymerization the tubulin protein disintegrates. That means they exist in dimeric forms, having two polypeptides, alpha tubulin and beta tubulin. These tubulin alpha and beta tubulin polymerization is broken down by colchicine, a cytotoxic material, chemical, and whenever we subject colchicine to the dividing cell, it does not divide further because the spindle fibers, astral fibers, they are made up of microtubules. Now come to the functions of the microtubules. They maintain the cell shape because they act as cytoskeleton, and the biconvex structure of RBC is maintained only by microtubules. Now, you might have studied Gaur system, that is membrane flow. Microtubules acts as muscles of the cells, and during exocytosis and endocytosis, the microtubules play a very important role. Movement of cilia and flagella, which also have microtubules, 9+2 arrangement, having 13 protofilaments. The axial support to them is provided by microtubules only, and their movement is caused by microtubules. The movement of chromosomes during cell division is also caused by microtubules because esters, centrioles, and spindle fibers are made up of nothing but microtubules. Their polymerization and depolymerization pulls the chromosomes towards the opposite poles, and thereby facilitating the cell division and equal distribution of chromosomes. Transport of cellular material is also caused by microtubules. In nerve cell, proteins transported down the axon, and structural support to the axon is provided by microtubules. In morphogenesis, also the microtubules are involved. The microtubule affects the change of cell shape during differentiation. Organelle formation is also aided by microtubules. Then, what are microfilaments? Microfilaments are actually actin molecules, and they are smallest structure, about 5 to 7 nanometer, sighted under electron microscope. They are found in clusters beneath the plasma membrane, and the cells which are non-muscular in nature, they show their muscular activity just because of actin and myosin present, interaction of actin and myosin present in them. The microfilaments, they are present, they are aligned in two different ways: parallel bundles. As I've told you in the beginning that microfilaments are actually actin filaments, and these are, you can see in the slide that actin subunit converts into actin filament, aided by adenosine triphosphate, and these then align into parallel bundles. Second one is loose meshwork. They are found in during the formation of pseudopodia. You can see the cross linkages of the actin molecules. Like colchicine, we have cytochalasin, which is a cytotoxic material. It blocks the polymerization and hampers the cytokinesis, phagocytosis, and amoeboid movement or change in shape of plasma membrane. That means that actin is actively involved in cytokinesis, phagocytosis, amoeboid movement, and in change of cell shape. Cytoplasmic streaming is caused by actin and myosin interaction. As you can see in the slide, the organelle is depicted in black color, and having myosin, myosin, as you know, having tail and head. Their head has ATPase activity, and when it binds to actin, the ATP is converted into ADP, and the organelle is moved on filament, actin filament. Myosin pulls the organelle around the tract of actin filament, and this is, as I have told you, is ATP driven. The tail region of myosin, as you can see in the slide, is bound to the organelle, and head moves along the actin filament. We also have intermediate filaments which are around 10 nanometer and have 2 alpha helical units, and there are around 45 odd intermediate filament genes present in human beings. Such the examples are keratin filaments, neurofilaments, glial filaments, heterogeneous filaments, and their deficiency disease is epidermolysis bullosa simplex. It is a muscular dysfunction, and also Meesmann's corneal dystrophy, and these two diseases are attributed to the intermediate filament and defect in the genes associated for formation of such proteins. So students, thank you so much